Dermomycoides, also known as Purkinjean clade organism, Purkinjean-like organism, is a protist parasite that is characterized as a spherical zoospore with a single flagellum, as you can see in the picture to the top right. It uses a single host life cycle, but can have additional transport hosts, and as a result of being able to use these transport hosts, has been documented across the United States and is known to infect a variety of amphibians, including the dusky gopher frog. Originally listed in 2001 as critically endangered, there are only about 200 adults in the wild population today. These animals will migrate into the pond to breed in the late winter and early spring, and their tadpoles will undergo metamorphosis until mid to late summer, where they emerge from the pond to find their permanent terrestrial burrow until they're ready to return for their first breeding event. Now, despite the work of conservationists and biologists and researchers, several years of low recruitment in the wild population has caused a shortened hydro period in the pond. Oh, fuck, I was supposed to, oh. Ooh, cool, cool, cool. All right. Thank you so much. Thank you for attending my talk. Dermomycoides, or Purkinjean clade organism, Purkinjean-like organism, or Purkinjesis, is a protist parasite that is characterized as a spherical zoospore with a single flagellum, as you can see in the picture to your top right. It uses a single host life cycle with additional transport hosts and has been documented across the United States in a variety of amphibian hosts, including the dusky gopher frog. Originally listed in 2001 as critically endangered, there are only about 200 adults in the wild population today. These adults will migrate into the pond to breed in the late winter and early spring, and their tadpoles will undergo metamorphosis and emerge from the pond to find their permanent terrestrial burrow in mid to late summer. However, despite the active work of researchers, conservationists, and biologists, several years of low recruitment in the wild population has severely hindered our ability to restore this charismatic species. And these years of low recruitment have been caused by a shortened hydro period where the pond dries before tadpoles are able to finish metamorphosis, or mass mortality events caused by infection of Dermomycoides. And when we look at the life cycles of these two species, it's really easy to see how this interaction can turn deadly. As adult dusky gopher frogs migrate into the pond to breed and deposit their egg masses, the tadpoles that result from these events are exposed to infectious zoospores of Dermomycoides that can penetrate and directly infect the tadpoles, and also non-infectious spores that are present in the water column that can hatch into infectious spores between a pH of 5.5 and 7. And while we know that not every tadpole that is infected with Dermomycoides dies, the vast majority do, to the point where we've had record years of breeding in the dusky gopher frog population, resulting in only at the most a dozen juvenile recruits. And so understanding what causes some of these individuals to survive while the vast majority die is a crucial step for the long-term recovery of this endangered species. And so my objective was to understand the effects of tadpole developmental stage on mortality of dusky gopher frog tadpoles exposed to Dermomycoides, so that I could help managers catch these mass mortality events before they occur and try and plan for them ahead of time. Because we are in South Mississippi, it is extremely hot and extremely humid. So unless you're there the day of or the day after a mass mortality event, you're likely not to see any evidence of it except for a single metamorph or juvenile frog that is dead. So understanding when these animals are at their most vulnerable is a key step to management of this disease. Now, prior work done by Matt Atkinson and Josh Cook, looking at tadpole developmental stage and infection of Dermomycoides, saw that there were changes across developmental stages and time of exposure when it came to survival and mortality with this disease. However, both researchers used different methods for characterizing the time at exposure. Josh Cook used hours after deposition in the pond, and Matt Atkinson used Gosner staging. And while both were extremely thorough in capturing a wide range of developmental stages and times, it does leave room for a knowledge gap. So what I did was I targeted my in order to bridge this knowledge gap, I targeted the developmental stages used in my experiment to capture the range used by both Josh Cook and Matt Atkinson using dusky gopher frogs. I exposed tadpoles as eggs, which is anywhere between Gosner stages 1 through 10, embryos, which is between Gosner stages 11 through 19, hatchlings, which is 20 to 22, and tadpoles 2 weeks post-hatching, which is around Gosner stage 25. I also had 2 control groups, one kept in the same room as those being experimentally exposed, and another kept in a separate quarantine room. Each treatment consisted of 10 individuals with 3 replicates, each representing a different egg mass, deposited in the same pond. At the time of collection from the pond, 3 additional eggs were taken from each mass for a baseline infection sample of Dermomycoides using qPCR. When the individuals developed to the stage needed for their treatment group, I exposed them to 50 zoospores per microliter of Dermomycoides in a small dish for 30 minutes, and then transported them to 2.5 liter aquaria after the exposure, where they were maintained and checked daily for morbidity and mortality. 2 weeks after their treatment's initial exposure, I collected a single individual for a midpoint infection sample, and 4 weeks after the initial exposure, I collected and killed all of the remaining tadpoles for endpoint infection level sampling. Now, when we look at the average days to mortality across these treatments, what we find is that there's a significant decrease in the time it takes for hatchlings and tadpoles 2 weeks post-hatching to experience mortality than it is for eggs and embryos, as well as our control groups. There was no significant difference between our control groups and tadpoles exposed as eggs, but as soon as they started elongating and became embryos, that's when there is a decrease in the average mortality. And when we look at cumulative mortality across all of the individuals by treatment group, what we see is that tadpoles exposed 2 weeks after hatching and tadpoles exposed as hatchlings both had a really dramatic mortality rate, in that over half of the individuals across replicate groups were killed by the midpoint sampling. And all of them were dead even before the endpoint. Now, tadpoles exposed when they're embryos survived up until the midpoint, but about a week before the endpoint of the experiment is when they started to experience mortality. And this is even seen in our eggs and control group. So what does this mean? Well, by combining the work that we've done with that of Josh Cook and Matt Atkinson, we see a fluctuation in survival across developmental stages. Josh Cook's work is in this dark blue, mine is in red, and Matt Atkinson's is in light blue. And as you can see, there's this really interesting fluctuation across developmental stages where the gopher frogs do seem to react similarly as the leopard frogs used by Cook and Atkinson. And one possible reason for these fluctuations across developmental stages is that those tadpoles that were exposed in their earliest developmental stages, so the eggs and embryos, they were protected by the jelly layer of the egg mass against accidental ingestion of spores of the water column and possibly even zoospores. And so that could have been what caused the lower mortality in those groups. Now, another possibility is that the immune system of ranid tadpoles is known to fluctuate throughout metamorphosis as the individual undergoes organogenesis. Both an immune response and organogenesis are highly metabolically taxing processes. So during the stage of hatching and 2 weeks post-hatching, when they're developing mouth parts and gills, this could be a stage where tadpoles are very vulnerable because all of their metabolic budget is going towards developing these organs and not mounting an immune response. And so the main implication that this has for managers is that we really need to target our surveys for those first 2 months after the initial breeding event for each of these groups, because that's when they're at their most vulnerable. You typically have tadpoles develop to Gosner stage 25 or hatch about 2 to 3 weeks after the initial breeding event. And so anywhere between those 2 weeks and 2 months, we need to see if we can monitor more often to catch these mortality events before they occur and really understand the environmental factors in the pond during that time. With that, I would like to thank our agency partners and I hope you have some questions.